Okay, so hello everyone, and thank you very much for joining. My name's Jian Liu, and I'm a senior research scientist from the University of Bristol. Today, we are going to explore an exciting area of research, cellular senescence and the emerging senotherapies for macular degeneration. This topic bridges basic biomedical science and a disease that impacts millions of people in the world, and I hope to share and discuss our recent understanding out and some advance for future treatment for this disease. We will begin with aging and what cellular senescence is, and then we will zoom into how it affects the retina and contributes to macular degeneration. Finally, we will talk about some exciting new therapies being developed to slow down or even stop the changes in the disease. In addition, I would also draw your attention to some of our recent publications in this area for your reference. During my talk, I will share with you some of the findings from our latest research. When we talk about aging, actually this term has two meanings, chronological aging and biological aging. Chronological age is just the number of years that have passed since our birth, but biological age is a different story. It reflects how our body is actually functioning. It's shaped by the gradual buildup of damage in our cells and tissues, and the slow decline in our body system. Although chronological age and biological age are correlated, they have different rates of change. For example, two people who are both 60 years old can have very different biological ages. The biological age can be affected by different factors. Chronological age itself is still one of the biggest risk factors for chronic disease and declines in our functions, including vision function, memory function, and motor functions. Family genetic background, for example, a person from a family with a mutation in a very important protein coding gene. The third one is environmental factor. For example, long-term air pollution can speed up the aging in the lung and in the airway system. Lifestyle choice plays a huge role in aging. For example, heavy drinking and smoking or eating a high-fat diet all can accelerate the aging process. Chronic health conditions such as type 2 diabetes, high blood pressure can speed up aging at the cellular level and also in the body. At last, the chronic stress, both psychological and physical, can significantly impact how fast our body wears down over time. By looking at all of these factors together, we can estimate our biological age, or in other words, how our body is acting from the inside. Just like our bodies age, so do our individual cells. And this process is called cellular senescence, which is one of the hidden processes in biological aging. Imagine a cell gets older and damaged. It's supposed to either repair itself or die normally, but sometimes it does neither. Instead, the cell may enter a state of permanent cell growth arrest or long dividing, but stays alive. This diagram helps us visualize the process and the stress from things like inflammation or DNA damage. A healthy cell can become a senescent cell, showing increased cell size and changes in the patterns of biological synthesis and energy generation. A key feature of these senescent cells is what we call the senescence associated secondary phenotype. This is a pool of inflammatory molecules that the senescent cells start to secrete. And this process is not always bad. In the short term, the process can be beneficial for things like tissue repair or wound healing. But when this process sustains over many years, they can become harmful, causing chronic inflammation in tissues and in organs, leading to age-related changes, leading to disease like macular degeneration. So how can we identify the senescent cells? We look for a set of biomarkers that can make senescent cells stand out. First, these cells have stopped dividing, usually because of DNA damage or stress from aging or harmful signals. Different from dead cells, the senescent cells stay alive, if not removed by immune system, which can cause problems over time. Inside the cells, they function differently. Their energy use and cleanup systems are off, and some important support proteins are missing in the cells. A flag marker is called senescence-associated beta-GA, which is an enzyme protein that builds up in the lysosomes. Lysosomes are special cellular organelles for cell recycling. Most importantly, as we just said in the last slide, they release a flood of inflammatory signals called the SASP factors, including different cytokines and growth factors, which can damage surrounding tissue. The unique combination of these features can help us identify senescent cells and makes them a primary target for new therapeutics. Our body has a natural process for cell death called apoptosis. So apoptosis is like a scheduled program to remove damaged cells from the body. However, this is not the case for senescent cells because the senescent cells have both death signal and survival signals at the same time. The survival signals allow them to resist apoptosis, which is why the cells can hang around in the body over a long time. The diagram on the left shows some important death signals and also survival signals with the most recognized BCL protein family. In our research, these survival pathways, known as SCARPs, are an ideal target for senolytic drugs, while inactivating these pathways to make the senescent cells sensitive again to death signals. We can go back a little bit to the SASP. Senescent cells are not just inactive. Actually, they are very active in communicating with their surrounding cells through a social life, while secreting a mixture of inflammatory molecules such as cytokines and growth factors. These SASP factors can target the senescent cells themselves to enhance the senescent state in the same cell, but they can also induce the changes in the nearby cells, driving them into senescence. The effects of SASP can be beneficial at short term, but they can be harmful at long term, improving or promoting the age-related changes and disease. And sometimes they can even promote cancer progression. Normally, our immune system acts like a cleanup crew, fighting and removing old and senescent cells before they cause big trouble. Different immune cells can play different roles in this process. Some immune cells destroy them directly. Some immune cells can eat and recycle the senescent cells, and some help alert the rest of the immune system. Senescent cells at the same time send out signals to say, come get me, or they can also put on a sign saying do not disturb to hide from the immune attack. When the immune system is healthy, this balance works well, but if the immune system weakens because of aging or disease, these senescent cells can build up, leading to ongoing inflammation and damage to the tissue. So here we can see age-related macular degeneration, or AMD. This disease is a leading cause of vision loss in people over 60. The diagram on the left shows a healthy retina. In the center is the macula. Macula is responsible to sharp central vision. In the middle, you see dry AMD, while yellow deposits called drusen build up and cause the retinal pigment epithelial cells, or RP cells, to die. The RP cells form a special layer just underneath the neuron cells in the retina. They play an essential role in feeding these neuron cells and keeping them healthy. When the RP cells are damaged, the neuron cells in the retina in turn slowly die, leading to gradual vision loss. On the right, we can see wet AMD, where abnormal blood vessels grow and leak blood or fluid into the retina. This will cause rapid and severe vision loss. From the picture on the right side, we see how vision is affected by AMD. A healthy macula gives you clear central vision. However, someone with AMD might see a blurry or dark spot in the center of the review. Here is how the story comes together in AMD. The slide shows images from donor eyes from different ages and conditions from people. And we used special stains to identify senescent cells in the tissue in the colors of green or red. You can see that the number of stained senescent cells is much higher in the older and AMD eyes, particularly in the RP layer. So the data from this study provides strong evidence that cellular senescence is indeed involved in the aging and also in AMD. To dig the science deeper, we looked at global gene expression in RP cells from patients at different stages of AMD. The circles on the left show that as the AMD progresses, there is a clear change in the expression level of genes associated with both cellular senescence and SASP. This data reinforces our hypothesis that senescent cells are not just a byproduct of aging, but are actively promoting the disease process in AMD. On the right, we have this table, and each row represents a special gene, and the colors shows which stages of AMD it changes in. So this analysis can help us pinpoint exactly which age-related genes are active at each stage of the disease. So this can guide us toward potential targets of AMD stage specific therapies. We used animal models in our study, And these mice showed signs of senescence in their retinal cells, just like in humans. And this model can give us opportunity to test mechanisms and therapies. On the left, we see images of the back of the mouse eye in a 5-month-old and a 21-month-old mouse, which is like an elderly person. The top images showed surface of the retina, and you can see little lesions in the young, but more lesions in the old mouse. The bottom images are cross sections of the retina, and again, you may see changes and damages in the old mouse. So this result tells us that aging causes visible damage to the retina. The top right panels show the signature of senescence in aged RP cells. The blue dots are the cell nuclei, and the green staining shows a mark of senescence. The older mouse has much more green spots, meaning there are more senescent RP. The bottom right graphs shows us that the mitochondria, which are the powerhouses of the cell, are not working well in the old mice. They showed low activity compared to the younger mice. This means that with age, the RP cells lose their energy producing ability, which contributes to the observed damage. So in short words, this slide demonstrates that aging leads to visible damage in the retina and an increase in senescent RP cells and a decrease in the energy production of these cells. Here, our research show the link of senescent RP cells and the inflammation. Think of it like a fire alarm system inside our cells. When our cells sense stress or damage from aging, the fire alarm goes off and it starts a chain reaction that causes inflammation. There is also a crucial off switch for this alarm, a protein called IRACM. Our data prove this in real life. The middle panels show that as humans get older, the cells have less of this important off switch, IRACM, compared to the young and the middle age. Without this protein, the fire alarm stays on all the time, as shown by other markers. The panel on the right shows that people with AMD have a lot of this fire alarm activity compared to the healthy eyes. So the key message here is that aging makes the cells in our eyes lose their ability to turn off inflammation. And this constant low-level inflammation is a major driver to age-related changes in AMD. The slide represents the emerging therapies to target senescent cells in retina. The idea is that as we get older, some of our eye cells become senescent. However, these cells release inflammatory signal proteins that fuel the progression of the disease. To fix this problem, we are developing a new class of drugs called senotherapeutics. There are two main types, senolytics and xenomorphics. Senolytics are like a smart cleaner that specifically finds out and destroys these senescent cells, removing the source of damage. Xenomorphics do not kill the old cells, but instead calm them down, preventing them from releasing these inflammatory signals in the first place. Here we show a xenomorphic treatment in the eye. We just talked about IRAK-chem being the off switch for inflammation. Our recent research used a gene therapy approach to deliver more IRAK-chem off switch to the eye. And the graphs on the left and in the middle show that this treatment effectively blocked the release of inflammatory signals and reduced the number of senescent cells in the tissue. The images on the right showed the real benefit. When we exposed mouse eyes to a damaging light, mimicking the kind of stress our eyes face every day, the eyes with IRAK-chem treatment were protected. There were far fewer dead cells and much less visible damage to the back of the eye compared to the untreated eyes. So in short, this data shows that by simply boosting our eyes' natural ability to control inflammation, we can prevent damage. So here we show our recent research testing a senolytic approach to treat senescent RP cells. So this figure shows a drug called ARV-825. This drug acts like a smart vacuum cleaner that can target old broken cells. It triggers a natural self-destruct process within the cell, removing it from the tissue. The top image on the right show our result. When the RP cells are exposed to stressors that makes the cells older, they turn blue as a sign of senescence. But when the drug is added, almost all the blue senescent cells were gone. What's even more exciting is shown in the bottom panels. Old cells stop reproducing, but this drug not only killed the old ones, it also rescued the remaining cells and allowed them to start dividing and reproducing again. So this treatment could be a powerful new way to actively clear out aged RP cells and boost the function of those remaining cells. In conclusion, let's look at the big picture. First, we have learned that old senescent cells are a normal part of aging, but they can cause chronic inflammation. And if this inflammation is more excessive, the process can contribute to disease progression, like AMD. The good news is that we are now able to precisely target these old cells. We can use two types of drugs, senolytics and xenomorphics, to combat the disease. By unlocking the science of cellular senescence, we can achieve a future of smart aging and strong vision for a better quality of life. At last, I'd like to take a moment to thank the many people and organizations who made our research possible. Our collaborators from universities in Bristol, London, Birmingham, and the US, Germany, and also Australia played an essential role in the collaborative study. We are also deeply grateful for the generous support from all our funding bodies, especially the Macular Society. Your continued contributions are essential in driving our research forward. Thank you all for your attention. And now I'm happy to take questions.
Thank you, Jian. That was absolutely fascinating. A really interesting topic for our audience. So I've got a few questions for you that came to mind as I was listening to you talk. Do the cells of the eye age more quickly than cells in the other parts of the body?
Thank you, Jerry. Yeah, that's a great question. So different cells in our body can age differently and with different speed. So in general, the eyes, the cells like RP cells and the photoreceptors, they age more quickly than other cells in the body. And the main reason is that because the frequent light absorption and conversion from the light signal to electric signal to the brain. For this reason, the RP cells and the photoreceptor cells have a very high demand of metabolic synthesis. So in that way, they age much quickly than other cells. So that's why the senescence takes a big part in the degeneration of the retina, particularly in RP cells and the photoreceptor cells.
Thank you. So I don't think you covered this. Why does the IRAC-M levels go down with age? Do we know?
Yeah, that's a great question. So it's the background is quite complicated, and basically the IRAC-M gene expression and the protein expression is under tight regulation at different levels by the cells. For example, we found that AP1 is downregulated with age, and AP1 is a regulator for IRAC-M expression. And with the downregulated AP1, and so IRAC-M is also, in consequence, is also downregulated. So that's one reason. And of course, there are other regulation mechanisms involved, like epigenetic regulation. So it's still ongoing.
Thank you. So the idea of biological age, I've heard about that before, but I don't know how would people find out or estimate their own biological age?
So some people can get blood samples. They can measure the epigenetic expression from the blood samples, and by that way, people can roughly estimate their biological age from the assay. So that's one thing, and also first we can also judge from the biological age according to their the combination affecting factors, as we said, like family, genetic background, and the lifestyle choices. From that, we can roughly estimate biological age of one given person.
And do you know, can you improve your biological age?
Yes, yeah.
Or?
Yes, yeah, that's right. So professionals develop drugs or therapy method to improve the aging and also treat the disease. But as individuals, what we can do is to get a good lifestyle choice. For example, no smoking can help us to be more healthier and slow down the aging process. And a good diet choice can also improve our health. Like if we can take more fruit and vegetable, this also help with health. So in other things like regular physical exercise, which can slow down our aging process and reduce the chronic inflammation and the senescence.
Thank you. And I'm sure something else lots of people are thinking. How close are we for a treatment involving IRAC-M?
So we have some promising results from laboratory in cell models and human stem cell involved RP cells. And we also performed animal experiments in mice. And next, we plan to use large animals like pigs and monkeys to test safety and efficacy. So we are doing these experiments to test the efficacy. And next, we hope to translate our results to clinical for clinical trials.
Excellent. We'll really look forward to reaching the clinical trial stages, and obviously be telling people all about that when it hopefully reaches that stage. So thank you very much, Jian, and thank you very much for that talk.
Thank you, Jerry.